Thank you very much for this nice introduction. So yeah, the aim of this talk today is to make an interactive actually seminar. So please do not hesitate to raise up your hand if you have questions or if I'm not clear. Really don't hesitate. And so I'm going to talk about immunotherapy because it's a therapeutical revolution, because now to treat cancer, we can treat actually the immune system and not specifically the cancer cells. So this talk will be subdivided in 3 parts. First, what is the immune system, then what is anti-tumor immunity, and then what is immunotherapy. So first, what is the role of the immune system is to fight against danger. So you have different sources that can be sensed as danger. So you have microorganism, injury, such as, for example, radiotherapy or just simply shocks. And you have also grafts when you have non-self cells that get into your self cells and then can lead to dangerous signal. And actually, each of these sources of danger is really various. And so, for example, microorganism, you have viruses, bacteria, parasites, and as you can see here, also the size is also very different. For example, a virus from nanometer, to nematode with 100 centimeter, for example. So in any way, these various sources of danger will be sensed by the immune system, and then this immune system will be activated in order to fight against the danger. But this activation of this immune system must be tightly regulated, because if you overactivate the immune system, then this can lead to autoimmunity. And so now, how does the immune system work? So actually, it's a really complicated networks of various organs. And in this organ, various cells, and in these cells, various molecules. And each this network is highly plastic and dynamic and coordinates. So, for example, the organization of the immune system is in different organs. So you have both primary lymphoid organs and secondary lymphoid organs where the immune cells are. So the aim of the primary lymphoid organ, so it's the bone marrow and the thymus, is to generate the immune cells. But at this stage, the immune cells will not be activated and mature, because they need to transit into this secondary lymphoid organ, and especially the spleen, in order to get activated, meaning that you have lots of immune cells that are here in your body, but will not be activated in order to avoid this phenomenon of autoimmunity. And only if you sense the danger, then the immune cells will get into your secondary lymphoid organs and then will be activated to fight this danger. And within each of these organs, you have various cells and that have a specific function. And basically, you can distinguish them between 2 groups: the cells that are aiming to work as an innate immune response, and the cells that trigger adaptive immune response. So in the innate immune response, you have monocytes, macrophage, neutrophils, dendritic cells, NK cells, so lots of different cells. And in the adaptive immune system, basically you have the T cells, 2 sorts of T cells, and B cells. So what happens when you sense a danger? First of all, you can have actually the no recognition specifically of the danger, because on your body, for example, the skin is a physical barrier, so it will impede the danger to get inside your body. So in this case, sometimes only physical barrier or even chemical barrier can lead to the stop of the danger, and then there will be no inflammatory response. But in most of the cases, the physical barrier or chemical barrier are not enough, and then the danger will get into your body, and then you will recognize the danger. And the first line of immunity to fight against the danger is the innate immunity. And this innate immunity is mediated by pattern recognition receptor. So what are they? So basically, the key source of danger are either pathogens or cells that are stressed. So all the different pathogens actually have these patterns that are shared between these different pathogens. And these pumps for pattern-associated molecular patterns will be sensed by the cells of the immune cells that have this receptor, so pattern recognition receptor. Moreover, cells that are stressed, so for example, a lack of oxygen, the lack of nutrients, radiotherapy, drugs can also stress the cells a lot. And also in this case, you will have damage-associated molecular patterns or dumped that will also activate the same cells with the same pattern recognition receptor. And so in both of these cases, you will have the sense of pathogen or stress cell that will lead to activation of the innate immune system. And that will lead to the huge release of molecules, so cytokines or chemokines that will attract de novo cells from the immune system. So first it will be innate immune cells again, in order to help them to work, because sometimes the cells that are on site are not enough. So this chemokine will lead to the recruitment of new cells from the innate immune system. And then also adaptive immune system. So in the case of an effective response only with these innate immune cells, actually the reaction will stop because there is no more sense of the danger. But if this response only with these innate immune cells is not effective, then the recruitment of all of these chemokines will lead to the recruitment of the adaptive immune response that will then act. And how does this adaptive immune response act? It's basically with antigen specificity. So what is antigen? So the definition is that it's a molecule that can initiate the production of antibody and causes an immune response. So here you have a pathogen that is represented by the letter 3, the number 3, sorry. And you can see that on this pathogen, you have lots of different motifs that are on this pathogen. And these different motifs actually are antigen because they can be sensed by antibodies. So the B cells are the main cells that will produce the antibodies. And if the antibody is specific to an antigen, so in this time the antibody number with the number 1 is specific to the antigen in number 2, then you will trigger adaptive immune response. But this response is highly specific because you can see here that the antibody will only target the antigen number 2, while the antigen number 4 that is also present in the pathogen will not be targeted by this antibody, maybe by another antibody that will be secreted by other B cells. Also, this antigen can be, for example, not only in microorganisms, they can also be presented by cells that are either in a stress condition or also can be infected, so the molecules of the pathogen can get inside the cells and then can be presented by the cells. And in this case, this specific antigen can also be sensed by the T cells that have the specific receptor called T cell receptor. So that's why we say that the adaptive immune response is highly specific, because these cells will only be effective towards a specific antigen. And also, this kind of adaptive response will lead to the expansion of these T cells and B cells, and also a memory response. So basically, how does the innate versus adaptive immune system work? So as I said, the innate immune system is really acts really fast. So you can have this activation within minutes or within hours, while you will need several days to have the recruitment of the adaptive immune system. The specificity is very limited for the innate immune system, because you have these molecules that are shared between these different pathogens or with stress cells, while the adaptive immune system will be activated only towards a specific antigen. Oh, yeah, someone I think want to get inside. The response to repeated infection is also not the same, because the innate immune system will always work in the same way, while the adaptive immune system will have a faster and more efficient way to fight against the danger because of this memory response. And so the key components are also not the same. In the innate immune system, you have all of the physical and chemical barriers, and the cells that belongs to the innate immune system that function with the pattern recognition receptor, while in the adaptive immune system, you have the T cells and the B cell that will have antigen specificity. And so, for example,
sorry, Laetitia.
Mm-hmm.
I just wanted to make a clarification. Could you go back to the last slide?
Yeah.
So this is Susanna, sorry. So I just wanted to emphasize for the people who don't know much about immunology, that one of the very key differences between the innate immune system and the adaptive immune system is that they both have recognition receptors, but pattern recognition receptors have been evolutionarily programmed, where every time we get infected with something, the bodies will say, oh, if we learn how to recognize this, then we can recognize it again in the future. And so the body responds better. And so we're more likely to pass those genes on to future generations. And so this is evolutionarily encoded based off of the microorganisms that we've been being infected with for extended periods of time, that we are consistently infected with. We learn how to recognize those and respond to them immediately, whereas the adaptive immune system is random. So it has a much higher specificity in terms of being able to recognize something because of the randomness of its generation, its production. And that means that it has to be controlled. So you need that innate immune system to control the adaptive immune system. Does that make sense?
Yeah. Thank you very much for your intervention, and please continue to do it. Can I continue? Okay.
Yes, please.
So here is the kinetics during a viral infection. So as you can see here, first you will have cells from the innate immunity that will be activated. For example, here NK cells with their respective cytokines like type 1 interferon. And then you will have the adaptive immunity that will increase through time, with this time it says the antibody response and also the cytotoxic T cells that will be activated. Okay, so now we briefly know how does the immune system work. Let's talk about anti-tumor immunity. So the concept of what is this concept of anti-tumor immunity? So as I started to explain, so the danger either come from the non-self, so either during a graft context or when you are exposed to pathogens, or the danger can also be come from your self cells in the context of stress. So the concept of anti-tumor immunity is that even if cancer cells are coming from you, so are considered as self cells, they are stressed because of this anarchic proliferation, and then in theory can lead to immune response. So this concept has been discussed by lots of scientists and since a very, very long time. And so first, Coley said that actually described the fact that there were more spontaneous regression of the tumor when the patient were subjected to skin infection. Then Elrich asked if the immune system can be involved in cancer initiation and evolution. But Burnett say that actually, if we have tumors, it means that the immune system doesn't work and doesn't sense the cancer cells. So for him, cancer cells are not targeted by the immune system. But then Thomas and Burnett said actually that T cells can actually recognize and target transformed cells such as cancer cells. And the real proof of concept came more than 100 years after with the parish that shows the proof of concept of anti-tumor immunity. So what did he do? So this team showed that actually if you treat mice with MCA, which is a component that is highly oncogenic. So here you can see the incidence of sarcoma through time. So if you inject this treatment in wild-type mice, so mice that are normal, or in specific mice, you have a different rate of incidence of sarcoma. So what are these RAG2 knockout mice? So these mice mean like they don't have the RAG2 gene. So basically, they will have no mature T cells and no mature B cells, meaning that they will not have effective adaptive immunity. So they will be immunocompromised. And so you see that here, these mice that are immunocompromised are much more sensitive to oncogenic agents in comparison to the control mice. And moreover, you can see here, it's the melanoma incidence in patients that are subjected or not to kidney transplantation. So you can see that patients subjected to kidney transplantation has an increased incidence in melanoma. So why? The immune system is implicated here is because when you're subjected to a kidney transplantation, then you will need to have some drugs that will slow down the immune system in order to let the graft within the host. So to tolerate this will slow down the immune system. And in the same case as the RAG mice that were kind of immunodeficient, you have an increased rate of cancer. So now that is the proof of concept have shown that the immune system can in a way control the incidence of cancer cells and then be kind of effective to target in the cancer cells. Why does the immune system can recognize the cancer cells? So here you have a normal cell that is completely self. And during the process of oncogenesis, the cells will have lots of different transformation due to mainly mutation, accumulation of mutations. And this will lead to a modified self that can be targeted by the immune system. So why? Because actually these damaged cells will have some mutation, as I started to tell you, and this mutation will lead to the formation of antigens. And as I said during the first part, this antigen can be sensed by the adaptive immune system. And so basically, the tumor infiltrating lymphocytes, so T lymphocytes that can actually invade the tumor, can recognize specifically this antigen and then recognize and then kill the cancer cells. So the concept is that the more you have mutations in your cancer cells, the more you will have the infiltration of this tumor infiltrating lymphocytes, and the better will be your anti-tumor response. So I have shown you that there is an increased incidence of cancer in immunocompromised patients in mice. As we have seen with pathogens, actually the cancer cells can also present antigen that will lead to the activation and amplification of the T cell that will then kill the cancer cells, that these T cells can infiltrate the tumor itself. But there is lots of other facts that have shown that actually the immune system interacts closely with the cancer cells. So now why cancer exists? Because if we have this anti-tumor immunity, it means that we can detect and kill the cancer cells. So we should not have cancer. So why do we still have cancer? It's because actually there are lots of different mechanisms triggered by the cancer cells that will make them invisible to the immune system. And this concept in another concept called immunoediting. So it's in 3 phases. So the first phase is the elimination phase, when actually the cancer cells that are just generated are detected by the immune system, and this immune system is highly effective and actually can kill the majority of the cancer cells. So actually, in your body, you can have lots of damaged cells that maybe will get into cancer cells, but your immune system will sense these cells and kill them. So actually, thanks to your immune system, you are avoiding lots of cancer within your body. But the problem is, like, sometimes the immune system is not effective towards all the cancer cells, and some cancer cells will actually escape and will be not subject to not be eliminated by the immune system. So you will have this equilibrium phase, while actually some cells will still be highly resistant to the immune system, while other cells can be targeted and killed by the cancer cells. So the equilibrium phase means just that you will have no growth evolution of your tumor, because some cell will persist but can be like in the dormant state, so will not actually divide, while some other will be targeted. But mainly when you have the equilibrium phase, some cells will still maintain their proliferative property. So in this case, you will have highly adaptive cancer cells that are that are not seen by the immune system, or worse, that induce the inhibition of the immune system, because they will produce lots of molecules that will dampen the immune system. And here you are in the escape phase, where when the tumor will grow. So the aim of the immunotherapy actually is to act on these different steps, and mainly during the escape phase, in order to activate the innate and adaptive immune response, in order to have this anti-tumor immunity. So the treatment option to fight cancer first is surgery. But if the cancer is disseminated, so at metastatic stage, you will not benefit from surgery, because it's completely impossible to target all of your lesions with the surgery. Actually, even if you can benefit from surgery, sometimes you will have a relapse, so the formation of a secondary tumor. And in both of these cases, patients that can't have surgery are patients with relapse. In this case, you will not have surgery anymore, and you will have drugs to try to treat cancer. So basically, first, it was chemotherapy that aims to target highly dividing cells. So that's why there is lots of side effects with these drugs. Then there were more specific drugs that this time target a specific feature on the cancer cells. So in this case, you will have less side effects, but the problem is that the cancer cells evolve so quick that you will have other molecular features that will not be targeted by this targeted therapy, and then you also have relapse. And with immunotherapy, what is kind of really revolutionary, actually to me, is that now to cure cancer, you will target the immune cells. You will not target the cancer cells. And you suppose that by targeting the immune system, actually, you will have a global response within all your body against the cancer cells. So there is different kind of immunotherapy. First, there is active immunotherapy that aims to strengthen your own immune system. So basically, it's a vaccination. You also have a passive strategy, meaning that this time it's not your own body that will produce all the molecules and cells that will fight against the cancer cells. It will be external components. So you have specific strategies. Basically, it's a cytokine that will just produce to boost actually a global immune response, but actually these cytokines are not really effective in anti-tumor field to treat cancer. But the specific therapies are kind of highly effective. So you can have cellular therapy. So for example, CAR T cells, meaning that it's T cells that are engineered in order to target specifically an antigen produced by your cancer cells. And this is highly effective drugs in leukemia, but it's highly expensive. So what is actually the most used is monoclonal antibodies that will specifically target a molecule. So how does a monoclonal antibody works? So either you have an antibody that will target specifically a molecule like as a receptor. And this antibody can be an agonist, meaning that when it will bind to the receptor, it will lead to the activation. And so in this case, you will have immunotherapy with agonists that will lead to the activation of the T cells. But and at the other side, you also have some molecules that will, when they will bind to the receptor, will lead to the suppressive effect on the T cells. And so if you have an antagonist, so that is immune checkpoint blockers, this antibody actually will take the place of the ligand that will that lead to the suppression of the T cells. And by doing this, actually, this antibody will then lead to the activation of the T cells. So you can see that you have various monoclonal antibody, either agonist and antagonist. And so the agonist will aim to activate the receptor that activate the T cells, while the immune checkpoint blockade will aim to inactivate the receptor that lead to the inactivation of the T cells, meaning that if you block a brake, then you have the activation of the T cell. And so these immune checkpoint blockades are the most broadly used in anti-cancer field. So in order to explain a little bit more how does immune checkpoint blockade work, I will do a little bit more of immunology, I'm sorry. So basically, during the previous part, I just said that the cancer cells can have this antigen that will be sensed by T cells when the T cells infiltrate the tumor in order to exhibit the cytotoxic activity of the T cells toward the cancer cells. But actually, it's much more complicated than this. So the first step is release of antigen by the cancer cells. And actually, these antigens here in the little red dots can be internalized by specific cells called antigen-presenting cells. And so these cells will eat lots of different antigen, and after they will transit to lymph nodes in order to specifically activate the T cells that have the specificity for this antigen that they present. And then these T cells, after that, will be activated, highly expand, and then go to the tumor through the blood vessels, and then will exhibit their cytotoxic activity. So the immune checkpoint blockade basically acts on this step 3, on step 6 and 7. So at step 3 is the antigen-presenting cells that will present the antigen to the T cells that have the specific receptor to sense the antigen. But actually, only this specificity is not enough to activate the cells. You also need the collaboration between the CD80 ligand from the antigen-presenting cells to the receptor CD28 of the T cells. While at the tumor state, only the T cell that is already activated will just need to sense the antigen presented by the cancer cells to exhibit this cytotoxicity. But at the stage of the lymph node, during this collaboration between the antigen-presenting cell and the T cells, the more activated will be the T cell, the more the T cell will express a receptor, and the receptor CTLA-4 actually will induce the stop of the activating signal, meaning that the cells will not be activated anymore and the cells will not divide anymore, meaning that the cells will not get into the tumor to exhibit the cytotoxic properties. While at the tumor site, the cancer cells that evolved can actually produce a ligand that will bind to the receptor PD-1 of the cytotoxic T cells. And this relationship will lead to the energy of the T cells, and that these T cells will not be effective. So the aim of the immunotherapy is to block this signal. So with anti-CTLA-4, you will block anti-CTLA-4, and then you will have a continuation of the activation of the T cells. While with anti-PD-1 and anti-PD-L1 immune checkpoint blockade, you will target this relationship in order to maintain the cytotoxicity of the T cells. So the first proof of concept that actually immunotherapy can work was made in ipilimumab with ipilimumab, so it's an anti-CTLA-4 antibody. And it was a huge revolution, because usually you have 0% of survival after stopping the treatment in metastatic melanoma. And thanks to this drug, you can see here that 20% of the patients with metastatic melanoma were still alive after the initiation of the treatment. So this was a really big revolution, because now we can say that with immunotherapy, actually, you can cure cancer. Because 10 years post-treatment still alive mean that you have no residual lesions, and then you have no tumors anymore. So you can, with therapy, you can just shift from metastatic stage, poor prognosis, and for this fraction of patients, they are completely cured. So this is, for me, a revolution. But sometimes you also have adverse events, immune-related adverse events, and especially colitis with anti-CTLA-4. And that's why there is the use of anti-PD-1 and anti-PD-L1 that are less toxic than anti-CTLA-4, and that also are highly effective. So here you have, the curve showing actually the survival of the patient during a clinical trial. So basically, when you start the clinical trial, all your patients are alive. And after you follow up, the survival of the patient after the initiation of the treatment, so either nivolumab here, that is an anti-PD-1, or a chemotherapy. And here, in still the metastatic melanoma, you can see that you have a clear benefit in increased overall survival thanks to anti-PD-1 treatment. This nivolumab can also be effective in other tumors, such as lung cancer. And here it's another study that shows actually the efficacy of, again, pembrolizumab over chemotherapy. And also, what is highly interesting with this kind of immune checkpoint blockade is actually you don't care anymore about the source of the cancer. Where is the cancer? What is the cancer type? So is it kidney cancer? Is it breast cancer? Because you activate all the immune system, and that can explain actually that anti-PD-1 or anti-PD-L1 treatment are highly effective in various cancers. Also, there could be a rationale to do combination with various immunotherapy. And actually, you can see here that when you treat patients with nivolumab and ipilimumab, you have an increased efficacy. But in this case, you can see that the improved efficacy is kind of moderate, while the generation of adverse events is really big, because you can see that, for example, with nivolumab, you have around 8% of the patients that will develop adverse events, with ipilimumab more, so 15%. But when you add ipilimumab to nivolumab, you have nearly 40% of the people that will have adverse events. So to conclude, immunotherapy is clearly a therapeutic revolution that can act on various tumors, that can lead to a long response. However, the toxicity can be big, actually, and have to be tightly monitored. And even if it's a therapeutic revolution, you still have a weak response rate, because most of the patients will not respond to immunotherapy. And so there is a high need to trying to understand why some patients will respond, why some patients will not. And so a huge need to do some research on this field. And so actually it's what I'm doing with lots of other scientists. And so some scientists are trying to develop other targets that can be blocked, for example, new immune checkpoint blockade, or other new agonists to activate the T cells. And other scientists are trying to develop biomarkers of immune checkpoint blockade response. So what is a biomarker? It's a biological molecule found either in the blood or the tissue that will follow up the disease. So actually you can have biomarker that will indicate a poor prognosis, a high risk group for a disease or serious side effect. But in the immune checkpoint blockade, what you want to see with this biomarker is either the patient will respond or not to this immune checkpoint blockade therapy. So today, there are mainly biomarkers that sense the tumor or sense the immune system. So the biomarker, for example, at the state of the cancer can be PD-L1. So you can assess to PD-L1 level in the tumor of the patient. And the rationale is quite simple. Because I have shown you that the cancer cells can have the upregulation of this PD-L1, and then when this PD-L1 can block the T cells. So actually, the more PD-L1 you will have, the more you will inhibit the T cells that are in the tumor microenvironment. So the more you will benefit from this treatment that will block this axis in order to reinvigorate the cytotoxic activity of the T cells. You can also assess the mutational load and how much tumor infiltrating lymphocytes you have within the tumor. So the concept also for this is simple. As I told you, the more antigen you will have that will be presented by the cancer cell, the more tumor infiltrating lymphocyte you will have, so the more anti-tumor immunity you will get. And on the top of that, if you increase the cytotoxic activity or you maintain the cytotoxic activity of the T cell, then the more anti-tumor immunity you will get. You can also have biomarker that will try to map the state of the innate or adaptive immune response. So actually here you can see that here it's the different cells that you have in the immune system, and here is the different cancer. And so you can see that lots of researchers are trying to find if these different cells are good actually or bad to induce immune checkpoint blockade response. So actually this kind of development of biomarker is highly ambitious, and actually lots of scientists are trying really to understand more what is doing each immune cells in these various cancer in order to understand how to improve a response rate to immune checkpoint blockade. And also the microbiome, so the bacterial flora that you have has been shown to also modulate immune checkpoint blockade response. And so basically, one biomarker might seem not to be enough to highly predict which patient will respond or not to immunotherapy. So now there is lots of scores that are being developed based on these different biomarkers that can be found. And basically, the combination then of this biomarker will much likely predict which patient will respond or not to immune checkpoint blockade. So with that, I would like to thank you for listening to this presentation. And I also want to thank Marie Nejon, that helped me a lot for this presentation, because we have made a course, and so basically I took sometimes some slides from her, and also by a render for all of these little cartoons. So please ask if you have any questions. Thank you.
Thank you very much, Dr. Nebot-Bral. That was a great presentation, packed full of information. Now we'll go into, oh, yeah, we see little claps. Yes, yes, thank you, thank you. We'll go into the next part of our event, which is the discussion. So please hop on the mic if you have any questions, or if you'd like to join any of the discussions that we're going to have. So I can start with a question. I might have missed this comment, but just wondering, so the monoclonal antibodies as an immunotherapy tool, since they target the T cells, can the same antibodies be used independently of the patient's type of cancer?
Yes, so this is you are a little bit freezing. I don't know. Can you hear me well?
Yeah.
Yes.
Okay, because okay. So yes, basically this is the aim. So basically the immune checkpoint blockade mainly targets receptors that are on the T cells, but you can also develop immune checkpoint blockade on macrophages or lots of different cells. And basically the aim is just to maintain the immune system active in order to have this cytotoxic activity. Do I answer well to your question?
Oh yes, absolutely, perfect. Thank you. We have Jota. Jota, want to come on?
Hello, thanks a lot for the great talk. It was really nice. Yeah, so then my question is a very specific point, but I got curious about it. For example, with the patients that got a transplant of an organ, like a kidney or something, are there already immunotherapies in the case for specific cancers? Like, I know the melanoma, like, can you treat melanoma without attacking the kidney that just came in?
So actually, it's a wonderful question, and it's a matter of lot of research. It's difficult to. So the first thing that scientists think is like, of course, you cannot give immunotherapy to patients with a graft, because then it will lead to the rejection of the graft. But actually, corticosteroids sometimes, so it's highly immunosuppressive drugs, can be used, for example, when you have big adverse events when you have immunotherapy. So in order to block the immune system, because in the case of toxicity with immunotherapy, you have a highly overactivated immune system. So you will have the injection of corticosteroid in order to stop this immune system. And actually, we have seen that when you give corticosteroid to a patient that is have this toxicity, then you will still have a response to immunotherapy. And actually, this kind of drugs, corticosteroid, can be are given or like brother or sister to corticosteroid, are given to patients that are subjected to a graft. So with that, there are lots of people that are wondering if actually we can give immunotherapy to patients that have grafts, even if they have drugs that are dampening the immune system. And it's clinical trials are ongoing, but I don't think that there is a clear answer to date. But it's a really, really good question.
Well, fingers crossed for the results. Thanks a lot.
Thanks, Jota. We have Gabriel.
Hello, really nice talk. Thank you very much. It's really hard to do outreach with this topic, but I think you did a great job. Thank you very much. My question is more almost a perspective from you as someone that's in the field and trying to think about the future of it. So of course, there's a lot of things being done, and just like specific questions trying to be answered in the ongoing field. But if you would, try to summarize how you see the field going towards, and the big questions, the big things that could be answered with what they are doing, and also the limitations, just like we can go into a certain point, but after that, I think it's going to be unreasonable and unclear. Could you just give your own perspective on that?
So actually, what the people are more focused in is now to develop new biomarkers. So with the new techniques today, we can assess the genetics of the tumors and the immunological profile of the tumor. And so basically, lots of people are trying to understand is like some mutational process will influence the response to immunotherapy, if the presence of these or these cells will also trigger or not a response to immunotherapy. And I think that with that, we will try to, predict better and better how improve this response rate to immune checkpoint blockade. But actually, the problem with this science is like it's really long and it's really expensive. And so I don't think that even if researchers are trying to understand more and more how to increase this response rate to immune checkpoint blockade, we can actually do this, because I don't think that in the clinic we can sequence all the time the tumor of a patient. We can do immunological profile of the tumor of a patient in order to understand when to treat, where to treat, with how many different combinations. So we are trying to do more and more stuff in order to help the understanding of how immunotherapy could work. But the major caveat is like the feasibility of this in the clinic, to me. Do I answer the question?
Any solutions to the problem?
To a solution to the
Hardcore
problem.
insights? Yes, I don't know. Maybe you thought about it, like how would you,
so
if you had a magic wand, what would you do?
So what I think is like now there is less invasive technique to try to assess to the immunological profile of the patient, and also the mutational landscape of the patient. Mainly now you see that you have what we call ctDNA. So it's DNA from the cancer cells that can actually transit in the blood. So with just a blood puncture, you can have this DNA, and that can from either the cancer cells or also in the blood, you have lots of immune cells. So just with this blood puncture, you can assess in a cost-effective way with targeting specific genes the cancer state, I would say, of like cancer profile of the patient. And you also have the immune cells, so you can just assess at that. And actually, I think that it's a good balance between easy and also sophisticated stuff that can be done fastly and also in a non-invasive fashion, because biopsy for a patient is also something to take into account. Okay. So I think all this liquid biopsy will be more and more and more investigated.
Wonderful. Thank you very much. Really fun.
Before we go to Susanna, just one bringing it down to a very basic question, because again, I don't work with this at all. So you mentioned the different tools that we have. So the cytokines, the cell therapy, the monoclonal antibody. I just wanted to confirm, are all of those delivered to patients through blood transfusion, or is there another?
Yeah. Yes. No, but no, vaccination is intradermal injection.
Okay, cool. All right, Susanna?
Hi. So I spoke earlier, but so my graduate work was partially in immunotherapy, but I'm now 3 years past when I got my PhD, and I haven't really been keeping up with the current immunotherapy techniques. So I know that when I was ending graduate school, rituximab, which is a monoclonal therapy for B-cell lymphoma, had kind of become a frontline therapy where they give it immediately upon diagnosis to certain people. But a lot of the checkpoint blockade therapies that you emphasize today are only given after failing chemotherapies, sometimes multiple times. So I was wondering about your thoughts as somebody who's still researching this and keeping up with the literature on giving checkpoint blockades early upon diagnosis versus do you think that the failure to respond to chemotherapies is something that's allowing the checkpoint blockades to work?
Okay, so this is a really good question. And I think to address this question, we just have to think a little bit of how the industry actually works. So the first thing is when you have like the easiest, I would say, strategy to develop a drug is first you try to put your drug when there is a high unmet need. And that's what happened with immunotherapy, and that was happened. Why the indication was at metastatic stage when actually there is no option left for these patients. So that's why historically, immune checkpoint blockade were given to patients that had no other options. And with that, we have seen that actually these drugs are really, really effective. And now, actually, immunotherapy is a standard of care for some of these diseases, this cancer, for example, in lung and melanoma, and for MSI tumor is the first of, is the standard of care. And so actually, with that, now that we know that immunotherapy can really be effective, now there are lots of clinical trials that show that immunotherapy actually can be effective at an earlier stage. And for example, you have neoadjuvant setting and adjuvant setting. So meaning that when the adjuvant setting is like you have the surgery of the patient, and usually after you give a drug. So now you have clinical trials that are assessing the efficacy of surgery, then treatment with immunotherapy or chemotherapy. And you also have neoadjuvant setting, where this time you give the drug, so either immunotherapy or chemotherapy, then you do the surgery. And actually, there is a big hope because, for example, for MSI tumor, so it's a kind of a tumor, with this neoadjuvant setting, actually, you had the complete regressions of the tumor after immunotherapy, meaning that this immunotherapy can be highly, highly, highly effective also at an earlier stage. So it's a big hope. And now actually, lots of clinical trials are trying to investigate immunotherapy in this earlier stage. And we all think that it actually is going to be highly effective.
That's so wonderful to hear. I'm really glad to hear that is finally moved forward. And especially going back to Gabriel's question, monoclonal is one of the cheaper forms of immunotherapy that you can have compared to the passive CAR T cells or the active forms with the vaccination with antigen presenting cells. Those cost hundreds of thousands of dollars per patient because it's truly personalized medicine. So if we can get these checkpoint blockade monoclonal antibody therapies to work, that is really the most cost-effective immunotherapy that we can do, and it's wonderful.
I completely agree.
And I assume as of right now, the burden of paying for all this falls on the patient, correct?
Can you repeat, please?
Just because Susanna was mentioning how expensive everything is and how it's like personalized medication. So I was just wondering, so is that covered by insurance usually? Like that the burden usually falls on the patient that's sick, correct?
Yes.
Yeah. Okay. So definitely focusing on the cheapest.
So the way that especially the cellular therapies that do have to be truly personalized, the initial patients that those were done on, they were done because of a grant. Basically, they enrolled in a clinical trial, and because they were in the clinical trial, they got it for free. But now that it's been proved that it works, it's no longer a clinical trial anymore. And so a lot of the money would have to come from the patient. The insurance company, at least in the US, is only going to pay so much. And especially if there's another treatment option that is 1,000 times cheaper for them, then they're going to have them go for that. They're not going to go straight to personalized therapies.
So that's why immunotherapy with immune checkpoint blockade is highly attractive, because it's much less expensive and can be highly effective.
Perfect. Any other questions, anyone? All right, everyone. This is it for today. Thank you very much, Dr. Laetitia, for your time and for sharing your knowledge with us. And to our audience, thank you for joining us either here live or watching this on YouTube later, if that's what you're doing. Kytos Seminar Series will continue in 2023, and we hope to see you again in the future. Susanna, did you raise your hand? No?
It was an accident. I was trying to press the clappy button.
All right. You can follow us on social media if you haven't already, and read about our projects and donate to organizations through our website. So have a lovely day, everyone.
Thank you. Bye.